Hi, my name is Brian Nelson. I'm from the Cobb County School District. I'm here today to talk to you a little bit about viruses. Let me start by asking you a question. Is a virus a living organism? Well, what do you think? Okay, let me give you a few clues. First of all, they don't breathe or use oxygen. They don't grow or get bigger. They can't reproduce on their own. And they don't respond to a stimuli. So now what do you say? Living or non-living? Well, the answer is they're non-living.
For such a big troublemaker, it's a remarkably tiny particle, about a millionth of an inch long. So small it can only be seen with an electron microscope. And it has an incredibly simple composition too. Single strands of RNA or DNA in protective layers of protein.
Viruses are pieces of bad news wrapped up in protein coats. They have the capacity to reproduce themselves. They do this by hijacking the machinery of the cell.
That is, the living cell that they infect. Without one to grab onto, viruses can't survive. As they reproduce, they destroy their host. No wonder the word pandemic strikes such fear in us. It represents a threat to us as a species.
Well, as you now know, a virus is a non-cellular particle. It's not really a cell. They're super small, have a protein coat around them with DNA on the inside, and they make their living, quote unquote, by invading actual living cells. There are lots of variety of viruses. They have many characteristics. We know they're microscopic, and besides having DNA, some viruses are even made of RNA. They have no true nucleus, even though they do have nucleic acid on the inside, and they can only reproduce inside of living organisms, which is why lots of times you can get a viral infection. They are host-specific, which means they only like to invade certain species. So a virus that your dog catches usually wouldn't want to invade you, and vice versa. So the good news, you can't catch a cold from your dog, and you can't give your dog a cold as well. In general, although we have seen some species of viruses that can cross over from animals to humans, like H1N1 that you may have heard of previously, or we believe the coronavirus may have switched over from animals to humans. Finally, viruses are not alive. As we discussed at the very beginning, they don't do all the things that living organisms can do. And so therefore, that's why they try to invade living organisms to help them reproduce. Now, viruses come in all types of shapes and sizes and can invade many different types of living organisms. One type of virus that you can see is the tobacco mosaic virus, for example. It actually invades plants, tobacco plants. You also have viruses that invade animal species, like the adenovirus and influenza viruses, as well as, believe it or not, viruses that actually will invade bacteria. They're called bacteriophages. Viruses, as I said, come in all types of shapes and sizes and can infect many different species. They're pretty crafty when it comes to reproduction. Two ways that viruses reproduce. One's called the lytic cycle and one's called the lysogenic cycle. We'll start with the lytic cycle. It's a pretty crazy cycle and one that bacteriophages are really known to do. What they do is inject their DNA into a host cell, cause the host cell to build more viruses, and then they explode out of that host cell in a process we call lysis to release more virus particles. Here, have a closer look. Here we see where the bacteriophage attaches to the bacteria, inserts its DNA, then it takes over the bacteria's machinery to make more virus parts. Finally, it builds and assembles those virus parts and then bursts, killing the cell. It's a pretty tough way to go. It's an interesting cycle. Check out this video to see more details.
The lytic cycle is the primary method of viral replication. and it results in the destruction, or lysis, of its host cell. A bacteriophage will attach to the cell's surface and release its genetic material into the host. Once inside the host cell, the viral genetic material will integrate with the host genetic material, utilizing it to express viral genes instead of bacterial genes. Consequently, large amounts of viral components, such as phage DNA and phage proteins, will produce in the host cell. Soon the phage components will be assembled as complete viruses, filling up the bacterial cell. Eventually, an enzyme will break down the bacterial cell wall. As the cell bursts open, the new viruses will be released. Summary: viruses will reproduce in bacterial host cells. During the lytic cycle, the viral components will destroy the host cell.
The second way that viruses can reproduce is called the lysogenic cycle. It's a little bit different from the lytic cycle. See, in this case, the virus, once it injects its DNA, it can form what's called a prophage or a provirus if we're talking about non-bacterial cells. And in that prophage or provirus, the virus DNA actually integrates itself into the host cell DNA. Here, have a closer look. Upon attachment and injection of the DNA, the virus DNA becomes part of the host cell DNA. What's really sneaky about this is that the virus DNA can stay in the host cell DNA while the host cell begins to divide. And then every time the host cell divides, it's also dividing with copies of the virus DNA inside of it. Whoa, pretty sneaky. Check out this clip for more details.
Lysogenic or temperate infection does not result in the lysis of the bacterial host cell. A bacteriophage will attach to the cell's surface and release its genetic material into the host. Once inside the host cell, the viral genetic material will integrate with the host genetic material, utilizing it to express viral genes instead of bacterial genes. As the bacterial cell replicates its chromosomal DNA, the viral component will also be replicated. Summary: viruses will reproduce in bacterial host cells. During the lysogenic cycle, the viral genetic material will integrate with a bacterial chromosome and replicate without destroying the host cell.
Viruses who are able to reproduce through the lysogenic cycle and who can incorporate their DNA into host cell DNA, they can do what we call laying dormant as that host cell divides. A great example of a virus that can lay dormant and that invades humans is the HIV virus. The HIV virus is one that's pretty dangerous because it targets your white blood cells. White blood cells are the ones responsible for fighting off infections. So if you have a virus fighting the very cells responsible for fighting things like viruses, it can put you in a pretty dangerous predicament. HIV uses what's called reverse transcription. It's pretty unique because inside of it, it has RNA instead of DNA. What the virus typically does is it injects the RNA into the host cell. It's one of the few viruses that has what we call an envelope, and that envelope is very similar to the host cell membrane. It allows the virus to get in to inject its RNA, and then using reverse transcription, it turns its RNA into DNA, and then that DNA can integrate itself into the host cell DNA. And then after a certain point in time, that virus can switch over to more of a lytic cycle and begin to have the host cell's machinery make more virus parts, eventually sending those virus parts budding out and taking a piece of that host cell membrane to form the virus envelope. Now, viruses, since they're not living and they can't move, have to have a way to get around. Viruses travel on what we call vectors. There are several viral vectors. People are a type of viral vector. When you sneeze or cough, like this guy, you can release all kinds of viral particles in the air and spread it to other people. Another way that viruses are spread is by touch, or by staying on objects until other organisms come around and touch that object. That's why we clean our countertops and tabletops and wash our hands really well, so that we can't leave viruses on objects for others to pick up later. Viruses can be airborne, which means they travel through the air. They can be waterborne, which means they're in our water sources, or they can be mosquito-borne. A virus famous for being transmitted through mosquitoes was the Zika virus. Throughout history, we've seen lots of epidemics and pandemics. An epidemic is a general outbreak of a virus. A pandemic is when it goes global. One of the worst viral pandemics we've ever seen was the Spanish flu of 1918. Over 500,000 people died in the US. That's a lot of people. That happened over the course of about just 25 weeks. And that's more people than AIDS killed in 25 years, more people than the Black Plague killed in 4 years. That's more people that even died in World War I. The Spanish flu is a pretty serious outbreak. Here, check out this video clip for more details.
1918 influenza was certainly the biggest, most devastating epidemic that we've had in modern times.
The worst human pandemic of the 20th century, the 1918 Spanish flu, broke out during World War I and spread swiftly around the world. Before it ended 18 months later, it had infected nearly 1/3 of the world's population and killed somewhere between 50 and 100 million people.
There were shortages of people in critical jobs. There were shortages of coffins.
The virus sparked when a bird flu mutated, becoming airborne, deadly, and highly contagious in humans. A subtype of the H1N1 family of viruses, the deadly flu stalked everyone, everywhere. There was no hiding place and no cure.
In 1918, there just wasn't the same kind of understanding of germs or medicines or therapeutics. It wasn't really there, and so there was lots of different attempts for herbal remedies, home remedies, anything that people could do.
But nothing worked. The flu disappeared in 1919 as mysteriously as it had arrived. It wasn't until 2005, when scientists cracked the code of the deadly 1918 flu virus, that the secrets of the pandemic began to emerge. With autopsy samples uncovered in old army medical records and a tissue sample extracted from the frozen body of a victim of the 1918 flu, microbiologist Terence Tumpey and a team of renowned scientists accomplished what was once thought to be impossible. They were able to first unravel and then rebuild the deadly strain, bringing it back to life.
We can only benefit from rescuing this virus and trying to understand why it was so lethal.
Here are a few more viruses that you may be familiar with. Smallpox is one you may have heard of. Ebola virus is one that broke out in the past, as well as the HIV virus. A class of virus that we're dealing with today is called the coronavirus. That's the virus class that's behind COVID-19, that's in the news, that's also causing the social distancing that we're experiencing today. But by seeing viruses of the past, like the Spanish flu, smallpox, Ebola, or even HIV, that we were able to survive, I'm hoping you'll have hope that we're going to be just fine with the coronavirus as well, all in due time. But to give you a little bit of information about the coronavirus and what it is, take a look at this video clip. If we're going to be able to protect ourselves from viruses such as the coronavirus, here are a few methods that scientists typically take. One thing that they may use are restriction enzymes. These are enzymes that are able to cut virus DNA. So when the virus injects its DNA into the host cell, those viruses will chop it up so that the viruses can't hijack the host cell's machinery and make more viruses. Another thing that's commonly used are vaccines. It's probably our biggest measure of protection against viruses once they strike. Vaccines are a weakened form of the virus that may also come with marker proteins from that virus. You see, your immune system operates by being able to recognize antigens when they attack your body. So if we can give your body the protein markers ahead of time, then your body already knows how to identify that virus when it sees it, and your body can begin making antibodies to fight off a virus if it ever attacks you. One more thing that we might use to protect us from viruses are to target viral enzymes. Earlier, I told you that HIV uses a process called reverse transcription to turn its RNA into DNA so it can get inside of our genome. Well, reverse transcription only happens using an enzyme called reverse transcriptase. So, scientists can target that enzyme so that the virus can never complete the process, reverse transcription. By targeting enzymes that viruses rely on, we're also able to protect ourselves from viruses. To learn more ways to protect yourselves from viruses, including things that you can do in your daily lives to protect yourself from the current coronavirus, visit the CDC's website at cdc.gov. So as you can see, viruses are definitely not living organisms, but they can sure wreak havoc inside of living organisms. I hope that you're more informed about viruses, and I hope you do visit the CDC's website to see more ways that you can protect yourself from viruses such as the coronavirus. Thanks for spending some time with me today while you're social distancing, and I hope to see you next time.